Welcome to the Transplantation online course from the Global Kidney Academy. My name is McKane and I'm a nephrologist in Sheffield. And the subject of this talk is antibody incompatible transplantation. Here's an overview of the talk. I'm going to describe the methods for measuring antibody and for cross-matching. I'm going to talk about methods for removing, modulating or inhibiting antibody. And then I'm going to describe the clinical aspects of both ABOI and HLA-I transplantation. And finally, I'm going to talk about live donor kidney sharing schemes. Now this lecture is longer than most in the online course, and I think it would be perfectly reasonable for candidates to divide it into two. And if you choose to do that, I would suggest you take a break after the first two sections. I'm going to spend quite a lot of time on the laboratory aspects because they're often poorly understood. The clinical aspects are quite easy to learn about from the published literature. So how do we measure antibody? Well, we need to be able to measure antibody against the isohemagglutinins, anti-A and anti-B. We need to be able to measure HLA-specific antibody. And in some circumstances, we may want to measure non-HLA antibody. I'm not going to talk about that. It's beyond the scope of this talk. It's something you may want to look into yourselves. And then we need to be able to do a final cross-match to make sure we're not going to get an early antibody-mediated rejection. And there are different techniques for doing this. So before we talk about measuring anti-A and anti-B, we need to know something about the antigen systems. So ABO or ABH antigens are really very different from HLA because they are carbohydrates, and those carbohydrates can either be in the form of glycolipid bound to a lipid molecule or glycoproteins. And the genetics are quite complicated and are encoded by the assembly enzymes, in other words, the enzymes that build these chains of sugars, and they're known as glycosyl transferases. Now, I can't go into the full detail, but this is a highly complex system. So, for example, there are subtypes of A, and you're probably aware of A1 and A2. In fact, it's far more complex than this. Furthermore, there are different core chains to these terminal oligosaccharides, and these core chains are differentially expressed in different tissues, and that can become important, and I'll illustrate this later. The carbohydrates can be in branching or non-branching chains, and because of the competing glycosyl transferases, the A antigen in AB individuals is not actually exactly the same as the A antigen in blood group A individuals. So here's a cartoon of the carbohydrate moiety of these molecules. So you can see that all of these molecules have this fucosyl side chain, an alpha-1,2 fucosyl side chain. And for blood group O, that's on the terminal saccharide. For blood group A and B, for blood group B, you've got another galactose residue here. For blood group A, you've got an N-acetyl galactosamine residue here. And then clearly if you've got AB, you've got both of these molecules. If any of you are interested in xenobiology, the principal xenoantigen on pig tissues is this molecule here without the alpha-1,2 fucosyl side chain. And there's quite a lot of cross-reactivity between xenoantibodies and anti-B. So A1 versus A2 is important to have some grasp of. So there are subtle differences in the structure of A1 and A2 and quite large differences in the antigen density. And in general, there's a lot less A2 than there is A1. And therefore, the antibody titers to A2 are typically lower. So the risk of rejection is less, and it's quite commonly possible to do a blood group incompatible transplantation A2 to O without any antibody removal. So you've got to be able to subtype A1 and A2. I talked about the core chains, and I'm not going to go through the details, but there are actually at least 6 core chains on which these terminal oligosaccharides are placed. And they've all got a slightly different structure, and they're not all expressed in different ways, in the same way in different tissues. And that can be relevant when it comes to antibody testing. So how do we measure isohemagglutinins? Well, the traditional methods are tube titrations or gel card titrations, but there are a lot of other methods in development because these are largely manual approaches. So some experimental systems describing ELISA, describing flow cytometry, where the antibodies are binding to either red cell targets or bead targets. Surface plasmon resonance, which is a very accurate way of measuring these antibodies, but probably largely a research technique. And thin layer chromatography to distinguish between antibodies binding to different core chains. So tube titrations is the classical method, and it's really not an automated technique, and there are a number of variations, but in essence you match recipient serum with donor red cells, or with red cells of a third party of the same blood group. You incubate in doubling dilutions in the tubes at room temperature, which is crudely considered to assess IgM, isohemagglutinins, and then at 37 degrees with an anti-human globulin, so like a Coombs test, and that's crudely considered to measure IgG. And the titer is the highest dilution where there's still agglutination, and that's measured manually. So you can see that there's likely to be some error. Gel cards, these are mainly the Diomed system. The gel card replaces the tube, but otherwise the technique is essentially the same, and it's generally considered that gel cards give slightly better reproducibility, and that's what most laboratories in the United Kingdom use. So there's a lot of error. There's inter-observer error, there's intra-observer error, there's the error introduced by using donor versus third-party red cells, and there's a degree of subjectivity about the cutoff. So this is really important because it means that titrations in one centre, the results, may not reflect the results in another. So the definition of a safe cutoff titer below which transplantation can proceed is a flawed one. So there are various studies. There's a Japanese study, an American study, and a European study looking at differences in tube titration results between different centres. This is the European one, looking at 3 centres who were using exactly the same overall protocol for blood group incompatible transplantation: in Stockholm, in Uppsala, and in Freiburg in Germany. And what you can see is that the range of titers for the same samples on these box and whisker plots for IgG and IgM are quite different. And another way of looking at this is the number of steps of difference. So 1 in 2 to 1 in 4 would be a 1-step difference. And you can see that the number of steps in difference really can be quite large, as many as 6-fold. I talked about core chains and how this can have an impact. Now we are measuring these antibodies using a red cell target, and red cells actually have most of the core chain antigens. So if an antibody binds exclusively to the type 1 core chain, that's only relevant if that type 1 core chain is in the donor organ. And in fact, in general, in kidneys and hearts, there isn't very much of the type 1 core chain. So you can get a circumstance, particularly after neonatal blood group incompatible cardiac transplantation, where you've got apparent antibody against blood group A, but where we now believe it probably isn't relevant to the transplanted organ. So that's enough about isohemagglutinins. Why do patients develop HLA-specific antibodies? And this is the term that I would encourage you to use. People loosely talk about DSA, but many of the HLA-specific antibodies we measure are not donor-specific. So you may develop HLA-specific antibodies after transplantation, after transfusion. Remember that white cells and platelets carry HLA molecules. Red cells don't carry many HLA molecules, but they do acquire some by passive transfer. They can't synthesize HLA molecules because they don't have a nucleus. Pregnancy, including early miscarriage. But it's important to realize that some of these new technologies, which we'll describe in a minute, are detecting very low levels of HLA-specific antibody, even in healthy non-transfused males. And we now believe that sometimes this is because the HLA proteins on the Luminex beads are denatured, and so you get false positives. And then the second question is, at what level of antibody binding do we call positive? And that isn't consistent between the publications. So how do we screen for these antibodies? Well, the traditional method was by CDC, where you have a cell panel of known HLA type that represents your donor pool. And that's where the phrase panel reactive antibodies, or PRA, comes from. So if someone has a PRA of 80%, it means that there's a positive reaction to 80% of the cells in the panel. Now, really, very few centres in developed countries are screening by CDC now, and so the whole concept of PRA is a bit flawed. I'll come back to that in a moment. And if you are screening by CDC, you need to make sure that you have a large panel that is representative of your donor population, and that's a lot of work. You can screen by ELISA, by flow cytometry, where the targets can either be cells, for example, Epstein-Barr virus transformed cells or CLL lines of known HLA type, or against beads. But really, most centres are using the multiplexing Luminex technology. And typically you have screening beads, which screen for class 1 and class 2, and then if they are positive, you can proceed to use the identification of phenotyping beads, which have a haplotype of HLA molecules in each bead, or the single antigen beads. And they allow a degree of quantification and some correlation with cross-match results and clinical outcomes. So here it is in a cartoon. In a CDC system, you mix serum of the recipient with cells of the donor, you add rabbit complement, and if there's cell death, then the vital dyes can discriminate live cells from dead cells. Now you can make this a more sensitive system by adding an anti-human globulin like a Coombs test. In a flow cytometric cross-match, you're not really interested in cell killing, you're just interested in binding. So again, recipient serum, donor cells, if there are HLA donor-specific antibodies, they'll bind to the cells, and you can detect these with fluorescent secondary antibodies, and you get a shift in the histogram here. And then the beads, where each bead has either a number of HLA molecules on it, or as I mentioned, for the most sophisticated method is with single antigen beads. Now I'm not going to read all this text out, you may want to pause and read it, but there's not really a huge amount of difference between single antigen beads in a Luminex machine and in a flow cytometer. A Luminex is a kind of flow cytometer, except that the process is much more rapid because it's a multiplex assay. So you're measuring lots of single antigens in the same run. And this is what you get from a Luminex machine. So for each individual bead, you get a mean fluorescence intensity. And most people consider a mean fluorescence intensity of 1,000 to be the cutoff between positive and negative. And you can see that there are degrees of positivity. And here tells you what antigen is on each bead, or if there's more than one in the phenotyping beads. Really, the message I'm trying to get across is that this is really complex to interpret, and HLA-specific antibodies may be donor-specific or non-donor-specific, but this is not necessarily a perfect distinction. So, for example, the lack of a donor-specific antibody in the serum is not the same as there being no antibody, because it may all be bound to the kidney. In general, you need a DSA to create a positive cross-match, unless there's a non-HLA antibody. Now, most systems will screen for A, B, DR, and DQ antibodies, and not everyone will screen for CW and DP, and we don't always know the DP type of the donor. Their significance is less clear, but they certainly can be pathologic. Now, the other thing to say about HLA-specific antibodies is that within a single antibody clone, you may have an antibody that will bind to many HLA molecules. That's because it's binding to what's known as a public epitope, so an amino acid motif that's present and available for binding on several different HLA molecules. Or you may get an antibody clone that will only bind to a single HLA molecule if the epitope is private. And so, if a patient has received a transplant mismatched to A2, they won't just develop antibodies against A2, they'll develop antibodies against a range of HLA molecules, even though they haven't been exposed to that HLA type of organ in the past. So the existence of these public epitopes leads to antibody clones that bind to common clusters of HLA molecules, and these are sometimes called cross-reactive groups, or CREGs. And in a research setting, it's now possible to define the amino acid motif, or eplet is the technical term, to which the clone binds. You need to have fully sequenced donor HLA type. You need to painstakingly correlate that with the Luminex single antigen bead results. And it's possible that that's the way antibody screening will go in the future. It's got a lot of quality control issues here. Until recently there were 2 manufacturers, although really largely one manufacturer dominates the market now. As I've mentioned, there are 3 levels of detection. There are the screening beads for class 1 and class 2, the phenotyping beads where you have an HLA haplotype on each bead, and then the single antigen beads. There are issues with batch variation, with interlaboratory variation, with the bead antigen density. So we know that the antigen density on the CW beads is not the same as the antigen density on the A and B beads, for example. And then, as mentioned, you can get non-physiological conformation of the HLA proteins. Now, I would encourage you to read this review, which describes all this in more detail. We tend to think that single antigen beads are always the best, and I think they probably are. However, there is some evidence to the contrary, and in this study from Zachary, you can see on the left here, using the phenotyping beads, they're the ones with a haplotype on each bead, you actually get a better correlation with the cross-match result than you do with the single antigen beads. I mentioned the phenomenon of false positives, so here are the data that this is derived from. So the first study was in Mexican male blood donors, 63% of whom appeared to have HLA-specific antibodies, mostly to very rare epitopes. And then a further study on male donors in cord blood, where again you see these natural HLA-specific antibodies, mainly to private epitopes. And often they're cryptic epitopes that are probably clinically irrelevant. So the single antigen beads are good, but they're not perfect. Now we tend to think of the mean fluorescent intensity as being an antibody quantitation measure, but in fact they're not licensed as a quantitative test. And there is a link between the titer of the antibodies and the MFI, but it's not a perfect one. I've talked about the definition of negative. Prozone describes a phenomenon whereby when you have very high levels of antibody, you actually get an increase in the MFI as you dilute the sample. There's some evidence to show that using single antigen beads you can see very rapid fluctuations in a single day. And when you have a strongly positive antibody clone that's binding to a public epitope, it will of course bind to many beads and that dilutes the effect. And finally, there are quite a lot of interfering molecules, biological agents that we use in transplantation, which affect the results. So really, I've talked about this already. This is data from Cambridge showing the correlation between the MFI to HLA-A beads in blue, HLA-B beads in green, and HLA-C beads in red, and the channel shift in the cross-match. And I've demonstrated there that I think that the relationship between the MFI and the likelihood of a cross-match for the C beads, for example, is extremely poor. This is data from Coventry, which is one of the centres in the UK that's done a lot of HLA incompatible transplantation. And again, you can see this phenomenon where there is a relationship, but not a perfect one, between the MFI and the strength of the cross-match. So these are patients with a positive CDC, so a very strongly positive cross-match, and in general the MFIs are 6,000 or above. These are patients with a flow positive cross-match, and in general the MFI is between 6,000 and 2,000. And these are people with a completely negative cross-match and generally fairly low level MFI. These are so-called bead positive transplants. And finally, it's very important that you've got good relationships between the laboratory scientists and the clinicians, because that's the only way you can make the most of this information. And I think this information has got to be available electronically because it's often quite difficult to integrate when you've got multiple paper reports. One thing I've not really talked about in detail is that not all HLA-specific antibodies are the same. Some of them seem better able to fix complement than others, and if they can fix complement, they're more likely to be pathogenic. So this is looking at graft survival in patients with no donor-specific antibody in black, with a donor-specific antibody which can't fix complement in blue, and then much poorer results in patients with donor-specific antibody that does fix complement. Now, I mentioned panel reactivity, and the fact that it refers to the proportion of cells in a CDC panel that react to a given serum. Now, the concept of a single PRA result is flawed because you will have a separate PRA for your class 1 panel and your class 2 panel, and you can describe the PRA for both the current or the historical serum. And as I've mentioned, you need to ensure that your panel reflects the donor population. Now, conventionally, we've defined sensitized patients as having a PRA of greater than 15% and highly sensitized as greater than 85%. I would like to discourage you from using the term PRA. It's really largely redundant in modern practice. We don't use CDC to screen for antibody anymore, and it's much more important to get precise identification of the antibody specificity. So what we've tended to move to now is what in the US they call a calculated PRA, and what in the UK we call a calculated reaction frequency. They mean roughly the same thing. So we use the Luminex single-antigen beads to define precisely the HLA-specific antibody. Then we predict the likelihood of a positive reaction based on a large pool of actual donors from the UK transplant registry, and it's typically a pool of 10,000 donors. And of course you can do this virtual calculated reaction frequency based on your current and historical serum, and you can integrate the results of class 1 and class 2. So you get a much better overall picture for a patient's transplantability. And there's an online calculator available, but it's not really any help to anyone outside the UK because it's based on our donor HLA frequencies. So why do we do a crossmatch? Well, we do a crossmatch to avoid hyperacute rejection and early antibody-mediated rejection. Increasingly, we actually do it to give us some prediction of risk. And it's not the same as HLA matching. So you could have a very poorly HLA matched donor and recipient combination with a negative crossmatch. And likewise, you could have a pretty well matched combination with a positive crossmatch because there's a donor-specific antibody to one of the mismatched antigens. So how do we do the crossmatch? Well, we mix the donor lymphocytes with recipient serum, and then we perform the assay in one of two ways, either by CDC or by flow cytometry. And we generally separate the results into class 1 T cells and B cells. You'll recall that T cells only express HLA class 1, and B cells express class 1 and class 2. There are a number of controls. Clearly there's a negative and positive control, and generally most centres will do an auto crossmatch now as well. Some centres will do an additional incubation with DTT to eliminate the IgM. And of course, now that we have accurate tissue typing and antibody screening, you could do a virtual crossmatch where you're doing a computer run to compare donor HLA type with recipient HLA-specific antibody spectrum. So for CDC, you purify the lymphocytes either from the lymph node of the spleen or the peripheral blood in living donors. You separate the T and B cells with magnetic beads, and you mix the recipient serum, treated by doubling dilution, under oil, 1,000 cells in a microliter using a Hamilton syringe. You add the rabbit complement, you add the vital dyes, ethidium bromide and acridine orange, and then you do a manual count of live and dead cells under a fluorescence microscope. And you can make the system more sensitive by adding the anti-human globulin, a practice common in the USA. So what you're measuring here is cytotoxicity, and you're measuring complement-fixing antibody. In a flow crossmatch, you're just measuring binding. So you're purifying the lymphocytes, you're mixing them with the recipient serum, you're adding secondary antibody labeled with fluorochromes to identify the anti-human IgG where it's bound, CD19 to allow you to identify B cells, and CD3 to identify T cells. You don't need to separate them magnetically, the sorter will do that. And then you assay the binding to T and B cells in the flow cytometer. And of course this makes no distinction between cytotoxic and non-cytotoxic antibodies, and it's only screening for IgG if you're using an anti-IgG secondary. and it's more sensitive than CDC. So to interpret the crossmatch, you require a knowledge of the clinical background. So this is a rather obvious example. If you've got a weak positive B-cell crossmatch in a non-sensitized male, then that probably doesn't mean very much. Whereas if you've got a weak B-cell crossmatch in a sensitized female, then that's a warning bell for you. And also remember that patients with autoimmune disease will often have an autopositive crossmatch as well as non-HLA antibodies. So conventionally, in the era before HLA incompatible transplants were being attempted, a positive T-cell crossmatch was considered an absolute contraindication. A positive B-cell crossmatch with a negative T-cell crossmatch, a relative contraindication. There's an assumption that this tends to be due to HLA class II antibody, and that's often the case, but it's not as straightforward as this. A positive T-cell crossmatch with a negative B-cell crossmatch is pretty unusual, and by definition this ought to be due to non-HLA, because there's no HLA molecules carried on the T-cell that are not also present on the B-cell. These rules are all becoming more blurred as we have superior antibody screening, and we need to integrate the crossmatch result with the clinical history and the antibody screening results. So I've mentioned virtual crossmatch. I'll just recap here. If you know the HLA specificities of the recipient serum and the HLA type of the donor, then you can predict the crossmatch result. And clearly there are computer algorithms for doing this. And you can use virtual cross-matching at several levels. So in the UK now, we've not been calling sensitized recipients for donors where we can predict a positive cross-match for many years. It's been part of our standard practice. Alternatively, if you've got a patient with a long history of completely negative antibody screening coming for their first transplant, you could argue that as the virtual cross-match is negative, you don't need any cross-match. And again, some centres are doing this in the UK to keep cold ischemic times down. Not many centres are brave enough to do virtual cross-matches for sensitized patients or to rely on them. And then finally, it's a very useful tool as a first screen for potential live donors, so that you don't need to get any donor cells. So what about removing antibody? Well, the principal way of removing antibody is plasma exchange, and there are different techniques which I'm going to run through with you because the literature can be confusing. Immunoabsorption is a more specific way of removing antibody, and that can be non-selective, in other words, just removing all immunoglobulin, or antigen-specific, just removing the immunoglobulin against the target that you wish to deplete. So this is a conventional straightforward plasma exchange circuit where we've got a blood pump, some anticoagulation, we've got a plasma separator. This is a filter-based system, but it could be a centrifugal-based system. And then the plasma is being removed and going to waste. And clearly, we've got to replace the blood volume with substitution fluid, typically a combination of human albumin solution and some fresh frozen plasma. So we could make this more sophisticated. We could separate the plasma, exactly the same as the first system, and then we could use a second filter, the cascade filter, to remove the high molecular weight substances and return the rest of the plasma back to the patient. Now, there are various reasons you might take this more sophisticated approach. You probably get more efficient immunoglobulin removal, but also you're returning useful proteins, non-immunoglobulins such as clotting factors, back to the patient. And you don't need as much substitution fluid. Finally, in an immunoabsorption circuit, you've got your plasma separator, could be a centrifugal separator, this is a filter-based one, and then you're running your plasma down your immunoabsorbent and returning that depleted plasma back to the patient. This is the TheraSorb system. This is an immunoabsorption system. Here the plasma separator is centrifugal, it's a microcentrifuge. And then you've got your separated plasma running down these columns, which are TheraSorb, which is a polyclonal anti-immunoglobulin. Now this column will soon become saturated, so then you switch to another column and you regenerate this column while this one is running. So in general, the more selective the system, the more antibody per plasma volume will be removed per plasma volume processed. And you'll tend to be more selective. Now, Glycorex, who make the blood group A and B columns will argue that by using antigen-specific removal, you'll get rid of less protective antibodies. You may allow treatment of patients with very high titers. But of course, there are downsides to this as well. There's a cost downside, and you also remove less complement, and it may actually be quite a good thing to be removing complement during plasma exchange prior to antibody incompatible transplantation. So again, this is data from Birmingham and Coventry looking at how much antibody is being removed per treatment. So this is conventional plasma exchange, this is cascade plasma exchange or double filtration, and this is immunoabsorption. This is a bit of a bullseye diagram describing the fact that the glycosauride system has antigen-specific removal, and so is the kind of best approach to removing anti-B and anti-A. But it's very important to remember that there are no head-to-head trials in this area. So what about modulating antibody? Well, we're really talking about intravenous immunoglobulin here, and I'm not going to go into any detail. I've put a good reference for this in the reading list, which describes all the mechanisms by which IVIG is thought to influence HLA-specific and anti-A and anti-B. So, and I've listed some of them here. I'm not going to go into any detail. I'd encourage you to read that review. Now, IVIG is often used in high doses at 2 grams per kilogram, often given over 5 days, usually without antibody removal and often with rituximab. Or perhaps more commonly now in transplantation at low dose, 0.1 gram per kilogram, after each antibody removal session. And there's 2 reasons you might want to give low dose IVIG after antibody removal. One is to replenish the protective antibody, and secondly because there may be a bit of a feedback loop operating here, whereby if you remove all the immunoglobulin, then you get upregulation of immunoglobulin synthesis. So what about immunosuppression? So really, most of the immunosuppressants we use will have some activity against humoral immunity, but we tend to particularly think about lymphodepleting approaches, rituximab and alemtuzumab. Now, although thymoglobulin doesn't deplete B cells, you will recall that to get class-switched IgG, you need T cell help. So depleting T cells will affect humoral immunity. Immunoglobulin we've talked about, and we'll talk a little bit about bortezomib and eculizumab. Neither of these drugs are licensed, of course, in transplantation. So this cartoon summarizes it, and you can read this review. So you could target the T cell or the antigen presenting cells, and really the common drugs that we do this with are steroids and thymoglobulin. And belatacept is not widely used, but certainly available now. We could deplete the B cells, but remember that CD20 is not expressed in the plasma cells, so rituximab doesn't deplete plasma cells. We could target the plasma cells with bortezomib. We could remove the antibody. Or we could say, well, we can't do much about the antibody, but we're going to affect its cytotoxicity by inhibiting complement with eculizumab, which is an anti-C5. And really, as I've described, IVIG has actions at all points on this pathway. Again, really just showing the same thing again. Rituximab is targeting the pre-B cell and the B cell and the memory B cell, whereas the bortezomib is targeting the plasma cells itself. You'll recall that bortezomib is a licensed treatment for multiple myeloma. I don't have much clinical evidence to show you here. It's mostly case reports and short case series, but here's a very small study comparing bortezomib with rituximab in acute antibody-mediated rejection, and a trend towards slightly better graft survival in the bortezomib group, but a very small study. Here's a very early case report describing the use of eculizumab as salvage treatment for severe antibody-mediated rejection. And you can see that the crossmatch results become strongly positive again here, and eculizumab has been added to the therapy, and that has rescued graft function as measured by urine output. So the Mayo Clinic group are the transplant team with the most experience with eculizumab, and they've made it a standard part of their HLA incompatible transplant program. And they continue eculizumab until the B cell flow crossmatch gets below a specific threshold. And by doing this, they've reduced the acute humoral rejection rate in their HLA-incompatible transplants from 40 to 7.7%. This is against historical controls. It's not a randomized controlled trial, but there are randomized controlled trials ongoing. And you can see that it doesn't affect C4D staining because you're inhibiting at C5, so later in the complement pathway, but you're really having a big impact on patients developing acute rejection, and therefore a great impact on the serum creatinine, and most importantly perhaps on the risk of transplant glomerulopathy at 1 year. So I think if you wanted to take a break from this lecture, now would be the time to do it. I'm going to talk about the more clinical aspects now, and I'm not going to go into as much detail as the literature is available, and I've included some of the important papers in the reading list. So in clinical practice, most centres doing ABO-incompatible transplantation are using rituximab induction, and the Swedish group at the Karolinska first presented data on this, almost describing rituximab as the wonder drug that made blood group incompatible transplantation feasible, but many people are not convinced that rituximab is essential to successfully perform transplants across blood group barriers. Some form of antibody removal using the GlycoRx antigen-specific system in the Swedish group, but plenty of other centres are using just plasmapheresis or double filtration plasmapheresis. And the GlycoRx columns are not licensed in the US. Now, as I've said, comparing titer cutoffs is a very dangerous game, but many centres would not attempt desensitization if the IgG titer is greater than 1 in 256. They might do with GlycoRx columns. And typically, most centres would be happy to proceed to transplantation once they had removed sufficient antibody to get the titer level down to less than or equal to 1 in 8. I prefer to see it at 1 in 4 or 1 in 2. And then almost all these programs have tacrolimus, MMF and prednisolone maintenance, although you will see some steroid-free regimes now. So I can't go through every regime, but this is a typical example. This is one of the other big US centres, Johns Hopkins. So they're starting the tacrolimus and mycophenolate 10 days before the transplant. This is true for ABO and HLA-incompatible transplants. They're doing alternate daily plasmapheresis with low-dose immunoglobulin. They're giving the anti-CD20 at the time of transplantation, along with the steroids and the anti-IL-2 receptor antibody. And then they're continuing with antibody removal after transplantation. In the Swedish group, they give the anti-CD20 a month before the transplant. And there are some theoretical reasons why you might want to do that. So what are the outcomes? Well, the Swedish group have been reporting equivalent outcomes to their ABO-compatible live donor program, but that isn't true in the UK, US and Japanese data sets. And all of these suggest that there's a modest excess of early graft loss, some of which is almost certainly immunological, but some of which may be technical. Antibody removal causes coagulopathy, and so there is a higher reoperation rate for bleeding. So here are the US data. I'm not going to go show you everyone's data, but you can see when you look at graft loss, there is just this early excess of graft losses in the incompatible group, and thereafter the lines run parallel. And the same is shown in the UK and in Japan. This is another important point. So the Japanese have been doing ABO-incompatible transplant in decent numbers for longer than Europe or the US. And this is important to understand that they are getting better at it, so the results are improving. So this is in the oldest era, the light blue, this is in an intermediate era, and this is in the most recent era. And in this review, they describe a sort of critical period after ABO-incompatible transplantation, sort of typically in the first 10 days when you can get aggressive humoral rejection and potential graft loss. But once you get beyond that critical period, these grafts do seem to behave much the same as any other transplants. And then another thing to remember, if you are doing blood group incompatible transplants and you have to biopsy these patients or you're doing protocol biopsies, is that C4D positivity is the norm. So seeing C4D staining in blood group incompatible transplants doesn't mean anything unless there's also evidence of microcirculation injury. So what happens to the antibody levels after the transplant? Well, after neonatal transplantation, you generally get very low or unrecordable levels of antibody against the isohemagglutinin, or the blood group sugar expressed on the transplanted organ. So this is a kind of peripheral tolerance. But you often do get some rebound in adults, and it's different in different series. The Swedish group tend to have reported very low levels of antibody rebound, or even none, whereas many of the other centres do report that you get low levels of antibody returning. So where you've got antibody that's returning, but the graft is stable, we term this accommodation. And I don't really have time to go into accommodation in detail, but it's believed that there's some form of adaptation of the endothelial cells of the transplanted organ so that they're resistant to antibody-mediated attack. What about the rejection risk? Well, roughly a 15% AMR risk after blood-group incompatible transplantation, and it seems to correlate better with the antibody rebound rather than the starting titer, but the literature are contradictory here. So what are the current controversies in blood-group incompatible transplantation? Well, there's a lot of uncertainty about whether rituximab is necessary and whether you ever need a splenectomy. It's a pretty rare event in most centres now. Do you need to remove antibody after the transplant? That was part of the standard protocol that the Swedish groups adopted, but many other centres only do antibody removal after transplantation if there's rapid rebound. And why is there this modest increase in early graft loss? Is it immunological? Is it technical? Is it a bit of both? So just to summarise the clinical aspects of ABO-incompatible transplantation, measuring anti-A and anti-B is not easily reproducible, and we hope that some of these new techniques on the horizon, such as surface plasma resonance, may help. We get good short and medium term results with acceptable levels of AMR and acceptable levels of chronic transplant glomerulopathy. But there still are areas of uncertainty. There are no head-to-head studies to establish what immunosuppression is best and what antibody removal technique is best. So finally, we're going to talk about HLA-incompatible transplantation in clinical practice. So first of all, why would we do this? We know this is high-risk transplantation. So typically these are patients who are obviously allosensitized and who've had very long waits on the deceased donor list. And their chances of a positive crossmatch with either a deceased or live donor are high. And these patients have got three options, and I think it is important to go through the options with them. In the UK, I would certainly be keen to offer them daily home hemodialysis, which actually may give a pretty good quality of life. They could wait patiently for a deceased donor kidney, but we know in patients with very high calculated reaction frequencies, if they don't get an offer in the first year on the list, they will probably never get one. Or, if they've got a live donor where the crossmatch is positive, they could go for an HLA-incompatible transplant, or they could go for a paired exchange scheme, which I'll talk about. This is UK data, audit data, showing that about a quarter of all live donor workups are halted by antibody incompatibility. So we're losing a lot of live donor transplants in the UK for this issue. So when are we going to do it? Well, it is a high-risk form of transplantation, so it's more likely it will be patients who are on dialysis rather than pre-dialysis patients. Typically after a long wait and where vascular access is precarious, so if you're running out of vascular access, you're prepared to take more risk. And also, in some centres, you're encouraged to have 1 or 2 cycles in a paired exchange scheme to see if you can get an offer before going down an HLA-incompatible transplant route. Now, at present in the UK, HLA-incompatible transplantation is only relevant for live donor transplants. Planned and emergency desensitization is possible in deceased donor transplantation, but it's very difficult to deliver. And really, the only publications describing this in the recent past are from Austria. So this is actually the most important slide of this bit of the talk. There's a spectrum of incompatibility. And it's really important when you're reading papers to try and understand how high risk the patients they were talking about. So you have a standard HLA-compatible transplant. The next step up is a patient where you can detect DSA by Luminex, but all the cross-matches are negative. So a modest increase in risk. Next step up are patients with a DSA who've got a positive flow cross-match, and finally the highest risk patients with a CDC positive crossmatch. So it's a hierarchy of risk, and as I mentioned, there's some correlation between the DSA strength. This is very like the Coventry data I showed you earlier. So these are the patients with the positive CDC crossmatch. They're not using MFI here, it's a slightly different measure. But you can see that the patients with the CDC crossmatch have the highest antibody levels, and then the patients with a negative flow crossmatch who are just bead positive have the lowest antibody levels. This is from the UK guidance on antibody incompatible transplantation, and again this is free to access on the British Transplantation Society website. And it's really showing the same thing, that if you're CDC positive with a high titer, then you're into this red, very high risk group. Where if you're just bead positive, your risk is only modestly above a negative cross-match transplant. And again, the relationship between the level of antibody and your risk of antibody-mediated rejection and your risk of having a positive CDC. But I think this graph oversimplifies it. So what could you do? And I think what you do depends on where you are on that hierarchy of risk. You could just do Luminex antibody monitoring and standard immunosuppression, and that's what some centres would do with a bead positive transplant. Or you could augment the induction and not do any antibody removal, so you might give thymoglobulin, for example, and there are other options. As I've mentioned, very few centres will do a splenectomy. Or you could modulate antibody with IVIG and or rituximab. And that's typically preparing people for deceased donor transplantation where the studies exist. Or in the high risk patients, you may want to do antibody removal before and after the transplant, and as I've mentioned, with low dose immunoglobulin. And I've talked about the different forms of antibody removal. You might want to do complement inhibition, but you'll need a lot of money if you want to do that because these are very expensive drugs. And you could inhibit antibody synthesis with bortezomib or even cyclophosphamide. So this is a French study from Denis Glotz looking at patients waiting for deceased donor transplants who were given high-dose intravenous immunoglobulin. And this was the impact it had on their panel reactivity. So it was improving their chances of being transplanted, either with a living or a deceased donor. And then this is a study from the US, from Stan Jordan, where high-dose intravenous immunoglobulin was given in combination with rituximab, and it's a very specific regime. And again, you saw improvement in the panel reactivity and an increased chance of transplantation. In fact, the Jordan group, previous to this, had done a randomized controlled trial, the reference for which I've given you, where they showed that high-dose IVIG led to an improved transplantation rate. And this is one of the few randomized controlled trials in the field. So I've described the Johns Hopkins regime to you already, and as I say, they use the same regime for HLA-I and ABI. So what does all this look like in practice? Well, this is data from Coventry. So this is what you get when you do the daily serial Luminex monitoring. So this patient has a donor-specific antibody to DR15, and then third-party non-donor-specific antibody to 4, 7, and 9. The antibody level wasn't particularly high here. The patient only had 5 plasma exchanges prior to transplantation, and the antibody levels came down to less than 1,000, which is the cutoff for positive versus negative. And then typically what you see for the first 5 days after transplantation is very low antibody levels in all these transplants, because if there is any antibody synthesis, it's all stuck on the graft. And then if you're going to get rebound, you typically get it around this 7-day level. And what you can see here is that the rebound of the third-party antibody is much higher than the DR rebound of the donor-specific antibody, possibly because much of it is stuck on the graft. So not many randomized controlled trials, but this is the Mayo Clinic effort to compare their results over different eras. So again, if you just give high-dose IVIG, you get DSA reduction, you get a proportion of patients, but not that high, to a negative cross-match, but you get a very high antibody-mediated rejection. At the other end of the spectrum, if you combine plasmapheresis, low-dose IVIG, rituximab, thymoglobulin induction, so that's very potent induction, and then aggressive antibody monitoring, you get up to CDC negative cross-match rates of 88%, but still an appreciable antibody-mediated rejection rate. This is from a review looking at antibody incompatible transplantation. And you can see most of these are relatively small series, but you can see that the antibody-mediated rejection rate is fairly consistent at around the 30% rate. And looking at 2-year death-sensitive graft survival rates of around 85%. So not fantastic results. This is the US outcomes, and as I said, you need to be really careful what you're talking about when you read these papers. So these are standard transplants. These are the patients who are just Luminex bead positive. These are the patients who are flow cytometric positive. And these are the patients with a positive CDC. And if we look at graft survival, you can see that actually the bead patient positives don't seem to do any worse. The numbers are small, but they don't seem to do any worse overall than the patients who are compatible. I've talked about the relationship between antibody level and outcome, and again, this is from Mayo Clinic. These are patients with a negative cross-match. These are patients who are bead positive. These are patients who are flow cross-match positive, and these are patients who are CDC positive, and you get more acute rejection as you move down that spectrum. So I would encourage you to read this paper carefully. This is the best medium-term outcome study in HLA-incompatible transplantation, and the authors are inclined to talk up the results, but I'm not so convinced. So it's a retrospective single-center review of 102 HLA-incompatible transplants and 204 matched controls. 5-year follow-up data with biopsies, not in all the patients, but in many of them. And to meet the entry criteria, they had to have a DSA, they had to start with a positive cross-match, and they had to get to a CDC negative and a flow cross-match of channel shift for B cells of less than 300 by the time of transplant. So it's a fairly well defined group. And the desensitization approach varied with era, as I've described for the Mayo Clinic already. So here are the results. So patient survival is not as good, as you can see, and that reaches statistical significance. Graft survival is not as good, as you can see. And you can see that probably the results for patients with class 2 antibody are significantly worse than those with class 1. And again, this phenomenon of the flow cross-match patients being better than the CDC positive cross-match patients. So what about the biopsy data? Well, this is where it all begins to unravel, because over 50% of surviving grafts by 5 years, where there was a biopsy, had transplant glomerulopathy. And we know that grafts with transplant glomerulopathy will have poor outcomes. So was the CDC a predictor of graft failure? Well, it seemed to be for class 1, but not so reliably for class 2, whereas the MFI and the Luminex was more helpful at predicting outcome for class 2. Now, I choose to cut these results a different way. By 5 years, 17% of the patients had died, 29% had death-sensitive graft failure, and 50% of what was left had transplant glomerulopathy. So you've only really got a third of patients without transplant glomerulopathy by 5 years. So why do you get so much transplant glomerulopathy? Well, it's the subject of another talk really, but when you have done a specific antibody, whether or not you've had an episode of acute antibody-mediated rejection, you will go on to get chronic endothelial cell injury with transplant glomerulopathy and proteinuria, and ultimately graft loss. So the enthusiasts for HLA-incompatible transplantation will cite this study, and it is an important study. So this is looking at the US in patients who were desensitized and had HLA-incompatible transplants versus equivalent patients who waited on the list for a deceased donor kidney. And that group are further subdivided into two because some of them received a transplant and some of them just remained on dialysis. So although the outcomes, and this is patient survival, so although the outcomes are not as good as a standard transplant, these authors would argue that if you go through desensitization, your overall patient survival will be improved. So what about paired exchange? Well, if you have an incompatible group in one centre and an incompatible group in another centre, and you can match them to get two compatible groups, then you convert two high-risk transplants into two low-risk transplants, and that's a highly attractive thing to do. And certainly in the UK, this is delivering growing numbers of transplants year on year. The blue bars here are non-directed altruistic donors, which is another subject entirely. And of course, you can have various levels of sophistication. You can just have two-way swaps, you can have three-way swaps, or you can use non-directed altruistic donors to kickstart donor chains. Now, of course, this can go wrong. If you've got a three-way swap, you just need one of these 6 patients to be unwell on the day, and the whole thing falls apart. There's a little bit of data from the UK system. So about a quarter of patients in the paired exchange scheme are proceeding to transplantation. And that's why some centres will say, we're going to do a couple of cycles in the paired exchange scheme, which runs every 3 months in the UK, before proceeding with an HLA incompatible transplant. And of course, you could try and integrate these two approaches. So you could take your positive cross-match and you could say, yes, this patient will be difficult to desensitize for a variety of reasons, therefore I'll go to a shared kidney scheme. Or you could take someone who's difficult to match but would be easy to desensitize for whatever reason, and then you might proceed directly to desensitization. And of course there might be people in the middle ground where you would try some cycles of paired exchange, and if that was unsuccessful, you would proceed to desensitization. Now, you can take this even further because you can say, well, we're not just going to use incompatible pairs, we're going to use compatible pairs. Why on earth would you do that? Well, this recipient here might, if they accepted the kidney not from their own donor but from another donor, might get a superior match. Or they might just be prepared to go into an exchange scheme for the greater good. This is just computer modelling, so it's not the real world, but you can see that if you don't include compatible pairs in your scheme, your transplantation rates will tend to sit at around 25%, exactly what the UK has demonstrated. But if you add in compatible pairs, either who will benefit, or even if they won't benefit, they're just doing it for the good of the service, then you'll actually get a much higher proportion of patients matched. We're not practicing this yet in the UK, but they are in some US centres. So the most important point is this concept of the spectrum of risk, that not all HLA incompatible transplants are the same. There's various ways you can approach HLA incompatibility, and there's really very few randomized controlled trials and no consensus. There is a high rate of acute antibody-mediated rejection despite desensitization, and high rates of chronic transplant glomerulopathy. And it's early days, but eculizumab may mitigate this. It's expensive, and there are other things to consider, in particular home hemodialysis, particularly daily hemodialysis, and paired exchange schemes. So it's been a long talk, this one, and if you have broken it into two, then I think that was sensible. But I think I've steered you through some of the pitfalls of measuring antibody, given you a better technical understanding of techniques for removing, modulating, or inhibiting antibody, and then a brief overview of the clinical aspects of antibody incompatible transplantation. For this lecture, I've done a very long reading list because it is a complex subject that will be new to many of you, and hopefully you'll enjoy reading this literature and grappling with the multiple choice questions. Many thanks.